In continuation to the previous lecture of enzyme function, today we will start clinical relevance of enzymes. Plasma enzyme assays can detect abnormal level of enzymes in plasma. Level may be abnormal due to from damaged tissue or lack of functional enzyme production. If level of an enzyme are raised, it may indicate damage to the tissue that enzyme is normally found in. If level are low, it may indicate that the enzyme is either non-functional, being produced slower or broken down quickly, possibly due to a genetic abnormality. This is a table which indicate if enzyme is lactate dehydrogenase. It found in everywhere. For example, liver, skeletal muscle, heart, kidneys, erythrocytes. And indication if it is raised is acute or chronic tissue damage. And degree of elevation can indicate extent of damage. If the enzyme is aspartate transaminase, it is widely distributed in liver, heart, skeletal muscle, kidney, brain, erythrocytes. And degree of elevation can indicate extent of damage. Indication if raised is liver disease, kidney failure, gallbladder disease. If the indication, if it is low, then diabetes, beriberi syndrome. If the enzyme is alanine transaminase, it is widely distributed but predominantly in liver. And the indication if it is raised is liver disease, viral hepatitis, bile duct problem. If the enzyme is alkaline phosphatase, location is found in greatest concentration in prostate. Specific isoenzyme in prostate, liver, bone, kidney, intestine and placenta. Indication if it is raised is prostate carcinoma. If the enzyme is creatine kinase, it is found in skeletal muscle, heart, brain, neurons, thyroid, kidney and intestine. And the indication if it is raised is skeletal muscle dystrophy and myocardial infarction in last 2 to 3 days and brain tumor. If the enzyme is amylase, it is found in saliva, exocrine, pancreas. And indication if raised is pancreatitis. If the enzyme is lipase, the location is exocrine pancreas and the indication if raised is pancreatitis. If the enzyme is acid phosphatase, its location is specific isoenzyme in liver, erythrocytes, platelets and bone. And indication if raised is diagnosis and treatment of prostate cancer. Now the reversible and irreversible inhibitors. Enzyme inhibitors are molecules or compounds that bind to enzymes and result in a decrease in their activity. An inhibitor can bind to an enzyme and stop a substrate from entering the enzyme's active site and prevent the enzyme from catalyzing a chemical reaction. There are two types of inhibitors. First is irreversible inhibitors and second is reversible inhibitors. Inhibitors can also be present naturally and can be involved in a metabolism regulation. For example, negative feedback caused by inhibitors can help maintain homeostasis in a cell. Other cellular enzyme inhibitors include protein that specifically bind to and inhibit an enzyme target. This is useful in eliminating harmful enzymes such as proteases. Example of enzymes inhibitors include poisons and many different type of drugs. Reversible inhibitors can bind to enzyme through weak non-covalent interactions such as ionic bond, hydrophobic interactions and hydrogen bonds. Because reversible inhibitors do not form any chemical bond or reaction with the enzyme, they are formed rapidly and can be easily removed. Thus, the enzyme and inhibitors complex is rapidly dissociated in contrast to reversible inhibition. For example, examples of reversible inhibition is, first is competitive inhibition, raised Km only. And uncompetitive inhibition in which the Vmx and Km values lowers, and non-competitive inhibition where only Vmx values lowers. Examples of irreversible inhibition. First is group specific, reaction only to certain chemical group, and second is reactive substrate analog, affinity label, inhibitor that are structurally similar to the substrate and will bind to active site. Third is mechanism based inhibitors, suicide inhibitors. Enzyme converts the inhibitor into a reactive form within active sites. Competitive inhibition can be overcome by increasing the concentration of substrate, while uncompetitive and non-competitive inhibition cannot.